So today I'm going to talk about the basic cell and the organelles, as well as some modifications made in plant cells so you can actually get photosynthesis there. Uh, but to begin with, let's start by looking at the basic cell. Now, all cell types, doesn't matter which type we're looking at, have a phospholipid bilayer, and all cells are alive. If we were to draw a phylogenetic tree, we would actually start with the archaebacteria, and somewhere along the line, some cells would develop a different type of ribosome. So a different type of ribosome, and we'd have bacteria here, but B for bacteria. Along the line, the archaea would actually continue on this way, and along the line, some cells would develop a nucleus. And the nucleus would actually end up giving us our eukaryotic cells. And eukaryotic continue to form a lot of different cells as well. Now remember, the bacteria didn't stop evolving, they continued and they made thousands of different versions. Archaea made their own, and eukaryotic made a lot. As we tend to deal with mostly with human biology, we will actually deal with that. But we do have protists, which are kind of a fallback category where it's every cell that is single cellular and doesn't fit into the other categories. We also have a group that actually develop a chitin cell wall, which would be our fungus. I'll put F for fungus. Then we have one that actually developed chloroplasm here and a cellular cell wall, which will give us our plants, PL for plants, and our animal cells continue here. So they're all alive. So eukaryotic cells all have a nucleus, and there are a lot of different types. You have protista, which are again the single cellular organisms. Some of these can do photosynthesis. Animalia, which are heterotrophs. Fungi, which are heterotrophs and have the chitin cell wall. And plant cells, which are autotrophs, which means they do have chlorophyll and have cellulose cell walls. They can perform photosynthesis, is what it tells us. Now, in the animal cell, we have a cell membrane, which I'll draw here. Cell membrane, phospholipid bilayer. We'll talk more about it later. You have cytoskeleton proteins. Remember, cyto means cell. So cytoskeleton proteins form the structure of a cell. We have enzymes in there, and some of the enzymes are ribosomes, which I always put as figure eights. We have a nucleus, which is another phospholipid bilayer with our genetic material in there. I'll just put a blue thing here for genetic material. We have something called an endoplasmic reticulum, which I'll talk about later. And we have some endoplasmic reticulum which has ribosomes on it, so it's rough endoplasmic and smooth. We have a Golgi apparatus which sorts the vacuoles, determines where they're supposed to go. We have vacuoles in here as well. And that's our basic cell. We have a lot of other things that happen, which I'll talk about later. Now, what I want to talk about first is enzymes, which are catalysts. And what a catalyst is something that speeds up a reaction but is not itself used. So enzymes are protein catalysts. That means that they can be, as they are proteins, they can be affected by pH and heat. Too much heat will denature the protein, meaning that it will change its natural form, making it not work. Too much pH can have the same effect, too little can also. And what they do is, as they're catalysts, they decrease the activation and energy of a reaction. A lot of times these can be paired to have two reactions, two things happening at the same time. Now, when we're dealing with enzymes, we have substrates, which are chemicals changed by a reaction. It involves enzymes, and a product, which is what you end up at the end of the reaction. So an example of this is if we have adenosine diphosphate and a phosphate, we can use an enzyme called, let's say, ATP synthase. ACE usually tells you it's an enzyme, and end up making ATP, adenosine triphosphate. Now what it does is it lowers the activation energy. So if we have a molecule, let's say we want to build something up, we have a low level of energy, we need to actually get a lot more energy, and that energy, part of that energy is going to go into the bonds, and the other energy is going to be lost, and we're going to end up having the energy that's in the bonds here. So we have a change in energy in the two structures. and we have a lot of energy that was lost. So what a catalyst does, it decreases the activation energy, so it makes it go a little faster. Again, enzymes are proteins, which means that they're encoded in our DNA, which, if you remember, our DNA is in our mitochondria. The DNA is copied into RNA, and the RNA is then copied by the ribosomes into an amino acid substructure, and it is bound together in a process called translation. One of the things with enzymes is they have a binding site for substrates, which means substrates will bind to an area, and when both the two substrates are together, it will actually cause the reactions to occur. By putting them that close, it ends up happening a little bit better. Now, for these substrates to bind to the reaction, it's dependent on a lot of different things. One is it's dependent on diffusion. And so if we increase the rate of diffusion, we increase how fast these substrates can bind to the binding sites. But again, if we have too much heat, we can denature the enzyme so it won't actually occur. It pretty much will stop functioning. Some things that can, there are some things that can bind to enzymes, causing a change in the binding site. So it's not actually bound to the binding site, it's bound somewhere else, and the binding site changes. And so what happens is it stops it stops the enzyme from functioning. These types of inhibitors are called non-competitive inhibitors. Sometimes things can bind directly to the binding site, causing it to compete with the substrate, which would be a competitive inhibitor. And these things will actually change how an enzyme functions. Different enzymes can be inhibited by different inhibitors. So, for instance, if we look at bacterial ribosomes, which we know are different than eukaryotic ribosomes, we can have some molecules that block the function of bacterial ribosomes, but don't necessarily block the function of eukaryotic ribosomes. Now, the reason this happens, the why, is because they're different structures. They actually have changed. The bacterial ribosomes are different, and so that's why you can have different molecules that will affect one type of ribosome but not the other. The reason this is useful is if you think about having a bacterial infection, we can give a medication that binds to bacterial ribosomes, inhibiting them, but not inhibiting the eukaryotic. So we can end up having medications that don't necessarily cause that. Now, another example of how we can change how these react is by having something like a selective serotonin reuptake inhibitor. So we can have a molecule that binds to something like a serotonin, which is a neurotransmitter, binding site, causing it not to be allowed to be brought in. There's more to the SSRIs, which you'll learn in pharm eventually. Some enzymes can work with similar molecules. So you have some enzymes called monoamine oxidases. These monoamine oxidases pretty much oxidize or break down monoamines such as serotonin, epinephrine. And so we can give inhibitors like monoamine oxidase inhibitors to inhibit the breakdown of a group of proteins. And so we end up keeping those molecules active, so they can bind to more of the receptors, the binding sites. If there are two enzymes that are needed for a reaction, sometimes we actually do have one reaction that happens by one, and then we have another reaction that happens by another enzyme. The slower of the two enzymes actually causes the reaction it actually will tell you how fast a reaction will occur. Now, if we look at alcohol dehydrogenase and aldehyde dehydrogenase, it's needed to take ethanol to carboxylic acid. And so alcohol dehydrogenase is a little faster than aldehyde dehydrogenase. So if you have too much aldehyde in the system, if you have too much alcohol in the system, you build up aldehyde, which will end up getting you sick. The aldehyde dehydrogenase is the rate limiting reaction because it's slower. Now we can use this in medication because we have something, for instance, like disulfiram. And disulfiram will inhibit aldehyde dehydrogenase, and so when you get this reaction, you'll build up aldehyde, causing people to get sick if they drink alcohol. Disulfiram is used as a medication to get people to feel sick whenever they drink, and so that's a medication used to pretty much help people get rid of their uh, alcohol abuse, but they have to know it's happening because if not, they can die. Now, the cell membrane, we usually refer to it as a fluid mosaic model. What it means is it's composed of many things. The things it's composed of is lipids, phospholipids, cholesterols, glycolipids. Glyco, remember, it means sugar, lipids, proteins, which can be transmembrane proteins, peripheral proteins, glycoproteins, sugars with proteins, lipoproteins, and they are also enzymes. Remember, enzymes are protein catalysts, and you can have some enzymes in the phospholipid bilayer. Now, one of the things that happens is you do have a special enzyme, sodium potassium ATPase, which pumps 3 sodium out for every 2 potassium. Sodium has a positive 1 charge, potassium has a positive 1 charge. So every time you're pumping 3 sodium out and 2 potassium in, you end up getting a net negative charge, which is seen on the membrane. Now, some membrane has an inner lipophilic barrier. The reason is, if you remember, the water-soluble heads will try to be on the outside where water is, and the fat-soluble will make a layer in the middle. So you have pretty much a lipophilic barrier. So fat-soluble molecules can cross, small molecules may pass through, but water-soluble and charged molecules need a protein channel to go through the cell membrane. Now, this is actually seen in osmosis and diffusion of things. So for instance, we have a cell here, and we have it in the same concentration of dissolved particles, solutes, inside the cell as outside. It will be an isotonic, and so the same amount of fluid will move in and out of the cell. Small molecules can actually move through here relatively easy, and so the water won't actually change the size of the shape. If, on the other hand, we have hypertonic solution, which we've talked about before, hyper means a lot more, or more, outside the cell, then it will actually drive the fluid to go into the cell, causing it to swell or even burst. If it's a hypotonic, it means that there's less solutions. If it's hypertonic, the water will actually be pulled out of the cell, causing it to crenate. And if it's hypotonic, the fluid outside the cell will move in, causing it to swell or burst. Hypertonic will have more molecules, so water will be pulled out. Isotonic, things will stay the same, and hypotonic, water will come into the cell, causing it to swell or burst. Now, inside a cell, we have what's called cytosol, which is a fluid inside the cell. Cyto, again, means fluid. The area where we find the fluid is called the cytoplasm. Plasma is a fluid mixed with different things, and this is a site where we have glycolysis. Now remember, glyco means sugar, lysis means break, so we're breaking sugar in glycolysis. This is an exothermic reaction, which means that we're going to get energy out of it, and when we get energy, we can use that energy to make ATP. Usually what happens is we put 2 phosphates onto glucose. There's a whole chain of reactions. At the end, we get 4 ATP back. So we put 2 ATP in, get 4 ATP out. So we end up getting 2 ATP from glycolysis. One thing to realize is it does not require oxygen. It doesn't use oxygen, so it's anaerobic. Now the cytoplasm is where all the organelles are held, and that's in the cell, so that's actually one of the things we're going to look at. Now when we want to break something down aerobically, meaning with oxygen, it needs to go, whatever we're going to break down, we need to get it into the mitochondria. Now before the mitochondria, sugar has to be broken down to pyruvate via glycolysis. We talked about it. And in glycolysis, we actually lose 3 hydrogens. They're still in the cytoplasm. They're used for other things. But we're going to end up seeing 3 hydrogens being lost. I have in the past shown you I have showed you the equation without it, but you do lose the 3 hydrogens. And so what you end up with is you end up with glucose, C6H12O6, going to 2 pyruvates here. You can see 3 carbons, 3 hydrogens, 3 oxygens, and the 3 hydrogens here are in the cytoplasm. Now, once you have pyruvate, that can be sent to the mitochondria, where it will eventually be broken down by oxidation. To do that, you actually have 3 more reactions. So there's 4 reactions to take sugar and get it into the mitochondria. But to get something in the mitochondria to be able to be broken, you have 3 reactions. The first one's called the bridge reaction, which no ATP is formed. Then there's a citric acid cycle where the ATP isn't formed directly. You do form GTP, and you form the products that are going to go to the electron transport chain where ATP is going to be formed. Now, if we talk about the bridge reaction first, pyruvate is converted to acetyl-CoA, and coenzyme A is pretty much partial enzyme that gets bound to acetyl, and to get that, you lose a carbon. So we end up going from 3 carbons to 2 carbons. Now, one thing to know is if you have too much acetyl-CoA, it can be used to form triglycerides, which will then be stored as fats and fatty acids, but we're going to talk about that later. So too much pyruvate, you end up making more fats. Acetyl-CoA is then shuttled to the mitochondrial matrix where you start having the citric acid cycle. It has a lot of different names. And what happens at first is the acetyl-CoA, which is a 2-carbon structure, binds to oxalic acetate, OAA, and it goes through the cycle. At the end, you release 2 carbon dioxides, which, if you remember, acetyl-CoA will have 2 carbons, and you make the oxalate again. What you end up with, though, is you end up with 3 NADHs. The N here is for niacinamide, which is a derivative from one of the B vitamins. You also make 1 GTP, which can actually be converted into ATP, and you make an FADH2, which is actually this is actually from it's another B vitamin derivative. And so what's going to happen is once you go through this, once you produce your 2 carbon dioxide molecules, the FADH and the FADH2 will be sent to, FADPH2 will be sent to the electron transport chain. And over there, the different things are going to happen. Now, I did mention here that if you have too much triglycerides, one of the things you can actually make them into fatty acids. If you have triglycerides and you want to use them to make energy, you can convert them to acetyl-CoA, and they can actually be metabolized through aerobic respiration. So when you want to break down fats, triglycerides and fats, you need aerobic respiration. You need to use the mitochondria, and usually to prime it, you do need some sugars to begin with. So going to the electron transport chain. So you have NAD and NADH2, and these are reduced to form NAD and FADH, which were used in the I want to use the same terms, in the citric acid cycle, and the hydrogens are bound to oxygens to form water. When you do that, you end up getting anywhere between 32 to 48 ATPs per glucose molecule. And so with this, you get a lot of ATP. Now, one thing I actually do want to tell you is sometimes the citric acid cycle is decoupled. It's not paired with the electron transport chain. And when that happens, you can't make the ATP. One of the things that can cause that to happen is something called cyanide, which is a poison. And if that breaks the linkage between the citric acid cycle and the electron transport chain, you have a rapid death because of the loss of the connection between the citric acid cycle and the electron transport chain. So you don't make the ATP, you don't get the energy, and cells die. Now, so far we've talked about the animal cells, and we have the cell membrane, which makes the outer border. We've talked about all the components of it. We've talked about the cytoplasm, which is where most enzymatic activities occur within the cell. We talked about the mitochondria, which uses oxygen to produce many ATPs. We've talked about the nucleus, which holds genetic material, which can be used to make ribosomes, which actually it can be used to make ribosomes, but the genetic material can be used to make RNA so that the ribosomes can make proteins. We have the cytoskeleton, which gives a cell its shape. Cyto, again, cell, skeleton, the inside structure. And there are a couple questions. What do we need to make lipids, not triglycerides, but lipids, and proteins that might be inside the cell membrane? And so for that, we actually can go to other things like the endoplasmic reticulum. If you break apart the word, endo means inside, plasmic is a fluid gel, and reticulum is a tubular structure. And you have two types. You have the smooth ER, which forms and breaks down lipids. And the reason it breaks down lipids is so it can end up going to the mitochondria. And the rough endoplasmic reticulum, which has ribosomes bound to it, so it can make proteins that are membrane bound or proteins that can potentially damage whatever's inside the cell, certain enzymes, for instance. We also have something called the Golgi, which sorts the vacuoles you make and determines whether they are leaving, they're coming in, or if they should be fused with other vacuoles, which we'll talk about a bit later. Now, there are some modifications to some bacteria, some protista, and some plant cells. So, for instance, one of the modifications is the things that allow it to do photosynthesis, which photo means light, synthesis means making, and it's a process of making sugars by taking light energy and binding carbon dioxide. There are some bacteria, which are called cyanobacteria, which can do this. There are some protists that have cytoplasm, chlorophylls, which can actually do that. We have plants that can do that. And what you have is you have two reactions when you do photosynthesis. We have the light dependent and the light independent. For the light dependent, what we do is we take carbon dioxide and water, and we use light to bind them together. And then we drive that to the independent reaction, where we take the carboxyl groups that we made and we bind them together to form sugar. And those sugars can actually be used to make a lot of different structures. Now we do need light to do this, and that ends up causing a decrease in carbon dioxide. As we have a decrease in carbon dioxide, if we have many of these, it does cause a cooling. If we start losing the plants, the protista, and the cyanobacteria that pull carbon dioxide out of the air, one of the problems is we end up increasing carbon dioxide, which causes a lot of problems. One of them is global warming. But if we don't want to talk about global warming, we can also talk about all the different effects that high levels of carbon dioxide has on respiratory system. For instance, the American Pediatric Association, the American Lung Association have all published a lot of articles showing how higher levels of carbon dioxide do lead to an increase in respiratory issues such as asthma, bronchitis, and decrease in overall health. Now, I do want to talk about mitosis, which is cell division. And cell division, what we're usually looking at is the DNA. Now, most of your cells, when you're living, are not going in cell division. Most of your cells are in something called interphase. Now, a long time ago, they thought that interphase was a time when cells were not doing anything. But in reality, that's when they're doing most of the stuff. Interphase is where most cell things are actually occurring. An interphase, if we are actually looking at the entire cell cycle, most of the cell cycle would be interphase. There would be a little part, which I didn't draw in here, of the cycle, where it would be a mitosis. Now, for mitosis, you can talk about interphase, which is before mitosis, and then we have prophase. Pro means before. So before we actually divide, prophase. Prophase, what ends up happening is we start forming what are called mitotic spindles, which are part of a cytoskeleton protein. And we also start breaking apart our nucleus. So we would have a nucleus which comes apart and our genetic material ends up just floating around in here. So prophase, we're making the mitotic spindles. Then we have anaphase. So then we have, prophase, then we have metaphase. And in metaphase, our centrioles grab our genetic material, our chromosomes, and line them up in the middle. And we call that area where we line up our chromosomes in the middle a metaphasic plate. So in metaphase, what we're doing is we're lining our genetic material in the middle of a cell with the centrioles holding them in place. Then we have anaphase. And I jumped the gun. Anaphase is where you start having the chromosomes being pulled to opposite ends of the cell. So we can call these poles, one pole over here, one pole here. So the centrioles, after they lined up our genetic material, they actually shorten and pull the genetic material aside. And they separate the genetic material. And as we separate our genetic material, we then end up trying to form a nucleus around it. And when we start forming a nucleus, up until we divide the cell, we end up in telophase. And so in telophase, we reform our nucleus, and we pull the cell in two directions. As the cells go in two directions, they split, giving you two daughter cells, but the genetic material is the same in both cells. So I actually used to tell people to remember, you have prophase, which is where you start breaking apart the nucleus and forming the mitotic spindles. Metaphase, where you take the genetic material and line them up into metaphasic plate. Anaphase, where you start pulling away the genetic material to opposite ends of the cell. And telophase, where the cell actually splits. And when the cell is splitting, we call it cytokinesis. Cyto means cell, kinesis means moving. So the two daughter cells pull away from each other. And so that's how we end up getting our basic mitosis, our basic cell divisions. To have this happen, we actually do have a lot of different things, but once it starts, we try to go through the entire aspect of mitosis. And you do have two parts of mitosis. You have mitosis 1 and mitosis 2. And what it does is it just splits the genetic material, so you only have two sets of chromosomes. Now, next time I will talk about meiosis, which is how you get daughter cells for sexual reproduction. And that's all I was going to mention today. I hope you enjoy the video and have a nice day.